what we're going to do is begin where we left off last time, and we're going to reverse transcribe this sequence into mRNA. That means we start at the 3' end of the template strand, and then the RNA synthesis starts with the 5' end. And we're just going to write down in terms of RNA what the sequence would be. If you haven't already done this, that's fine. We'll just do it together. It's pretty quick. Every time there's a T, you put an A. Every time there's a C, you put a G, and so on. You got to watch for the A's, because on the template strand where it's an A, in RNA it's going to be a U. If you go to look for T's in the genetic code, they're not there, and that's because all RNA has U's instead of T's. So let's just do this as we go along. Catch me if I make a mistake, which I'm likely to do. A string of T's here is, oops, that should be A's, right? Then C, G, C, A, U, G. And why are we doing this? Because we could just ask a computer to do this for you, but you'll have something like this on an exam, perhaps not this, and perhaps not this long of a sequence, but I want you to at least have practice for what we're doing. That can't be right. And as you're seeing, as I go through this kind of quickly, it's really easy to make mistakes when you're going through it kind of quickly. So it may be worth your time, if you're doing this on an exam, to go through it once, try to get it as close as you can, and then check it again. Just make sure you've got the right sequence. And again, one easy check to make is if your RNA sequence has any T's in it, you've done something wrong. Okay, that's the RNA sequence 5' to 3'. Now, if we were going to sort of do this for real, if you were doing this on an exam or on a problem set for a shorter time, I would recommend that you write out the mRNA sequence from 5' to 3', and then that you divide it up into 3-letter codons. If you want, we can do that. That's just a lot of letter writing. But on the other hand, if we do boring stuff now, we would have to do less content-rich stuff later. So let's do the write the letters. Okay. This is like doing worksheets or centers in elementary school. This is not as fun as centers. My kids, was that a thing when you were in elementary school, doing centers? My kids used to love it. That's where, anyway, you go to different desks and you do different activities. All right, well, all right, I'll get started, and what I'm going to do is write out the sequence 5' to 3', and I'm going to draw little lines between each of the codons, and we're going to begin with AUG. That is our start codon, and that's what actually defines a gene, or what's called an open reading frame in an mRNA sequence, is the start codon. So when you see that AUG, that's where you begin, and then you can write down the codons left to right, 5' to 3', and then it'll be a little bit easier to get the genetic code. And if you want to divide your sequence up above into those sort of segments so it's easier to see. And remember, you're reading it in this direction, so you've got to read backwards. UG. There's an UG. All right, so now we're continuing on, testing our ability to read backwards. Let's see, ACC, AAC, GCG. So now, if you're ever caught on a long plane flight, just print out for yourself some mRNA sequences and the genetic code. And instead of singing 99 bottles of beer on the wall, you can do this. Okay. Well, there's my sequence. I'm not sure if I've made any mistakes or not. I know what this amino acid sequence should be, so we'll just go along. How are you doing? Have you got it written out? You're faster than me. Not surprised. Okay, so start codon AUG. That one you can just memorize if you want. That's always methionine. And here's your genetic code down here, and you don't have to, your codon table, and you don't have to memorize the codon table. You can always just use it. So AAA is, we decided, which amino acid? That's lysine. There was some kind of attempted mnemonic device for that last time that I can't remember what it was, so well, it must not have been useful. CUG is leucine. CCG is what? Proline. Okay. There's another CCG, so that's easy. What about GGC? GGC is glycine. Good. UGG. That's tryptophan. Then GA or GA. GAA is glutamic acid. AAA is lysine again. CGC, let's look for that one. Arginine. AUG is another methionine. AGC is serine. And then CGC we just did, which was arginine. AGC we just did, which was serine, so 2 serines in a row. GGC we already did as well, and that was glycine. CGC is another arginine. Then GUG, G-U-G is valine. Then UAU is tyrosine. I found it interesting that, and then UUU is phenylalanine. I found it interesting that tyrosine and phenylalanine are chemically similar. They're both benzene ring side chains. And it's interesting to me that, well, and then tyrosine has the additional OH group. It's interesting to me that the one base pair change, A to U, doesn't generate a dramatic change in the character of the side chain, which is, again, probably useful from an evolutionary standpoint if you can allow individual one base mutations to not dramatically change the protein product. That's probably a pretty good advantage. Okay, AAC is N, asparagine. CAU is histidine. AUU, isoleucine. ACC is threonine. Then AAC, again, I think we've seen that one before. That was asparagine. GCG is alanine. AGC is serine. CAG, I forget, that's glutamine, Q. Then UGG is W. GAA, we've had that before, that's glutamate. CGC we've had before, but I keep forgetting it, it's arginine. CCG, it's proline. AGC is serine. GGU, almost there, glycine, and UGA is stop. So there you go. It turns out this is the sequence from a protein called, a segment of a protein called PIN for proline isomerase, and this is called the WW domain of proline isomerase because this segment has two tryptophans. You wouldn't be expected to predict this, but this sequence folds into a triple-stranded hairpin with two turns, and it looks kind of like this, only it's twisted. And I did a lot of work with this protein in my postdoctoral work and in my early research when I came to BYU. Okay, so that's the exercise, just sort of decoding things. Any questions about how to do that? We could then ask the question, what is this thing's net charge at pH 7 or pH 2? So we would first remember that the N-terminus is positively charged and the C-terminus is negatively charged. And then we would go through and find all the lysine residues, that's a plus, and the arginine residues. So pluses on lysine, arginine, continuing to scan the sequence. There's another arginine, nothing there, and then there's one more arginine. Any others that I missed that you guys can see? No. Okay, and then all the glutamates, E's and D's. I only see two glutamates in this sequence. So total 1, 2, 3, 4, 5, 6, 7 pluses. And 3 minuses. So overall, that net charge should be 4 plus at pH 7. If we were at pH 2, all the carboxylates should be neutral, and then histidine would be positively charged. So at pH 2, it would be 8 plus. All right, so now you know how to do translation of an mRNA sequence to form a protein. But we haven't talked at all about how that process happens. And importantly, we haven't talked at all about where this genetic code comes from. Because there's no sheet of paper like this that the ribosome looks at while it's looking at an mRNA sequence and then says, oh, right, that one's leucine. Let me open up my drawer. Here's a leucine. All right, there you go. It's more complicated than that, but more automated than that and more beautiful than that. So to understand the genetic code and how protein synthesis happens, we need to understand something called tRNA. All right, so T stands for transfer. It's transfer RNA. And what is it supposed to transfer? It's supposed to transfer an amino acid to the right position in protein synthesis. So we've thought of mRNA as just this single-stranded piece of DNA that is just a sentence of individual codons. And that's not entirely accurate. The mRNA does fold up into specific structures. But the tRNA in particular, the shape of the tRNA is important. This is impossible to draw for you, so I'm instead going to rely on figures from the notes that I took and prepared, and then we'll talk through sort of a little bit more about the tRNA structure. So I'm just going to copy this. And we'll paste it here. So this is the sequence of a tRNA from yeast. And it's a tRNA that is specific for phenylalanine. How can you tell that it is specific for phenylalanine? Because there's a segment right here on the tRNA, which we are labeling and highlighting in red. This is the anticodon. The anticodon exists in a loop, and it is complementary to the codon in the mRNA. So if you look at your genetic code, one of the two codons for phenylalanine is UUG. So I've displayed that codon from the 5' on the right to the 3' on the left in an existing segment of tRNA, of mRNA. The anticodon loop of a phenylalanine tRNA matches and is complementary to the codon in mRNA. Yeah? Why didn't you ask? I should have, thank you. Just, that was me failing to pay attention till the end of the stuff I was writing. I sometimes do that with writing. I also sometimes do that while speaking. It results in embarrassing situations. So there you go. This is the phenylalanine codon. Therefore, the anticodon is from 5' to 3' is GAA. Some other features of the tRNA. This is how people try to represent two-dimensionally what three-dimensional tRNA structure is. This is the 5' end of the tRNA. This is the 3' end. Places where there are lines represent Watson-Crick pairs across a single strand. So one of the neat things about tRNA is that a single RNA can actually fold back on itself and form localized double helices. So for example, this segment GCGG forms a double helix with a corresponding segment UCGC in a later part of the tRNA strand. They call these things, they call these segments stems. And if you look over at this structure of a tRNA, you can see locations of some of these stems. Anytime it looks like double-stranded DNA twisted, that's one of these stems. So the sequence goes here, through another stem to a loop, back through that same stem, through another stem to the anticodon loop, back through that same stem. There's a little bit of a loop here, another stem, another loop, back through the same stem, and to the 3' end. And at the 3' end, there's an overhang. And the 3' hydroxyl, this is where an amino acid is going to get attached to the tRNA. So here's the structure of the tRNA. You draw it out two-dimensionally, it looks like some kind of T or cross. You look at it three-dimensionally, it no longer looks like a T or cross. It's sort of folded back in on itself. It's mostly L-shaped. And if you want to remember that tRNAs are L-shaped, that's fine. The anticodon, but you don't write it like an L shape, you write it like a gamma or an upside down L-shaped. Or, yeah, anyway. The anticodon is down here. These three bases are the anticodon for where the tRNA binds to mRNA. And then this portion here in yellow is the overhang where the free 3' hydroxyl is, and that's where the amino acid is going to be attached. So importantly, a tRNA has two key places: the anticodon that recognizes mRNA, and then it has the acceptor hydroxyl where the correct amino acid needs to be attached. Okay, does that make sense? Now, all of this is great. For every codon in that genetic table, you have a tRNA that has the corresponding anticodon. That's more or less true. It's more complicated than that, but we're just going to go with that summary. Now, the question that you could ask is, okay, I understand how the tRNA can be complementary to the mRNA codon through base pairing. Does that part make sense? Yeah, hydrogen bonding between the bases. But how does the tRNA know which amino acid should be attached to it, and how does that amino acid get attached? So there's the second key to the genetic code is a class of enzymes that are called aminoacyl tRNA synthetases. And these are specific. They recognize the right tRNA, and they recognize the right amino acid, and then they bring them together. They load the amino acid, or some people will say charge. They charge the amino acid, the tRNA, with the amino acid. We won't use charge because that's going to be confusing. Loading the amino acid onto the tRNA. And the other thing is that these synthetases are ATP dependent. They're going to consume ATP in order to load the correct amino acid on the correct tRNA. And that's actually sort of reassuring. This is a key portion of information transfer in biology. You've got to get the right amino acid on the right tRNA. And so it makes sense that energy should be required to store and process that information. Okay, so we're not going to talk about at a molecular level what's going on when an aminoacyl tRNA synthetase binds the, say, phenylalanine tRNA and then binds phenylalanine. We're not going to talk about that molecular recognition. That's something that people have studied. I just don't know a lot about it. So you'll have to trust that the enzyme has evolved to specifically bind the correct tRNA and the correct amino acid. That's not trivial. That's very difficult to do, but it's amazing that it works. Okay? Is that summary level okay? I know that some of you can be unsatisfied. Well, how does that, I know, you just have to look it up and learn it later. I'm not going to be the one that teaches that to you. Okay, but you can understand the basic mechanism for how an amino acid is loaded. It's loaded in two phases. You're going to take the amino acid that you're interested in, and since we've been dealing with a phenylalanine tRNA, let's just draw phenylalanine. So step one will be the enzyme takes phenylalanine, and it's going to use ATP pyrophosphate is going to be released. I'm not going to have enough space over to the left, so I'm going to do this. And what you're going to make is called an aminoacyl AMP. So this is going to track very similarly to how we did carboxylic fatty acid activation at the just before beta oxidation. If you recall, we used the carboxylate on the fatty acid as a nucleophile to attack the alpha phosphorus of ATP, thereby generating fatty acyl AMP. This is exactly the same chemistry. The carboxylate of the amino acid is going to act as a nucleophile to attack the gamma phosphate, or sorry, the alpha phosphate of ATP, and then you're going to get pyrophosphate as a leaving group. Just to help you follow along, this is the gamma I'm getting it wrong again. This is the alpha phosphoryl group of ATP. That's the one that got attacked by the carboxylate. Here's the phosphoryl phosphorus and the phosphoryl oxygen. How would you show the mechanism of this reaction, of this first step in the aminoacyl tRNA synthetase? This is the amino acid. I'm just abbreviating the carboxylate. And here is ATP. This is the gamma phosphate group. This is the beta phosphate group. This is the alpha phosphate group. And then this is the ribose ring. And we'll abbreviate the adenine base. This is the alpha phosphorus. That's going to be the electrophilic one for this enzyme. And all of this over here is the pyrophosphate leaving group, which is sometimes abbreviated in biology as PPI. It's just a really good leaving group. So the oxygen of the carboxylate attacks the phosphorus. You're going to get a pentavalent intermediate that looks like this, where the amino acid carboxylate is attached to the phosphorus. Then you have here the pyrophosphate leaving group, which I'm just going to abbreviate like this. And then over here you have, well, I wanted to abbreviate, but we'll do the ribose ring, and then the adenine base. Electrons on the phosphorus kick down, and pyrophosphate leaves, and that's going to get you to your product. So no surprises there. Basically the same mechanism you saw before in activation of a fatty acid carboxylate. The effect of that is to convert our amino acid. It used to be a carboxylate, which is not an electrophile, but the effect of this reaction, when you have this aminoacyl AMP, is suddenly the carbonyl carbon of your carboxylate is itself attached to a good leaving group. That is the beauty of the acyl phosphate. From one perspective, the phosphorus is electrophilic and the carboxylate's a leaving group. But from the other perspective, the carboxylic acid, the carbonyl carbon is electrophilic, and then all of this is the AMP leaving group. And that's important if we want to attach the carboxylic, the carbonyl carbon of our amino acid. If we want to attach that to tRNA, we've got to first turn the carbonyl carbon into something that's got a good leaving group. Does that make sense? We start with a carboxylate, we put the AMP on there, now the carboxylate's attached to a good leaving group. Now we're ready to charge or attach the amino acid to the tRNA. So this is, we'll call this amino acid activation. Okay, questions about that mechanism? So second step is just exactly what you would expect. We would call this basically nucleophilic acyl substitution. You're going to take the fatty acyl AMP and the tRNA, which I'm going to represent like this, squiggly, 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 and 3' hydroxyl group. And afterwards, we're going to get AMP as the leaving group. And then you're going to get squiggly, 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 3' hydroxyl group now in an ester with the amino acid. Okay? How does that happen? That part's pretty easy. A base in the enzyme deprotonates the 3' hydroxyl group and it attacks the carbonyl carbon, breaking the pi bond, getting a tetrahedral intermediate. So we'll show that tetrahedral intermediate here. Negatively charged oxygen. I'm going to abbreviate AMP as this. It's the oxygen from the alpha phosphate. It's basically everything that's green above. Is that okay? We could draw it out if we wanted to, but since we're tired, let's just save ourselves some pain. And then here is the tRNA, and it just needs to sort of look like an L. Then electrons on the negatively charged oxygen will be, which is going to act as the kicker-offer. Those kick down and AMP leaves, and you end up with the tRNA, and this is now called an amino acyl tRNA because the tRNA is charged or loaded with an amino acid. Here, the tRNA 3' hydroxyl group was the nucleophile. And now the amino acid is loaded onto that hydroxyl group. The bond we formed is an ester bond. The bond we broke was an acyl phosphate bond. So that's going from level 1 down to level 2. So this reaction is thermodynamically favorable and downhill. And that's important because the tRNA, now that it's loaded with that amino acid, has to swim over to the ribosome, and you don't want that bond hydrolyzing in the meantime. We've already invested some ATP to get us to this point. It would be sad if water came in and hydrolyzed that bond. So we replace the acyl phosphate bond in AMP with a more stable ester in the amino acyl tRNA. And that's going to have a long enough lifetime to get to the ribosome and do what it needs to do. Okay, questions about that? I think those mechanisms we just drew, I can't remember what I said on your quiz, but that may be the second question on your quiz. And if you've drawn it in your notes, we'll call that good. Okay, questions about that? Okay, should we talk about the ribosome? So we're going to use mostly cartoons for this because the ribosome is enormous. But in your lifetime, the structure of the ribosome was determined at atomic resolution. So if you want to now, you can go to the internet and go to the protein data bank and say, show me the structure of the ribosome. I want to see all the atoms, and it will appear. And you can look at it, and there are many, many structures of the ribosome now available at various stages of protein synthesis. So people have learned a lot about how protein synthesis occurs. You could spend a couple of weeks talking about it, or perhaps longer. I'm not prepared to give you more than just a really brief summary. The ribosome has a couple of subunits. So there's a larger subunit and a smaller subunit. And the mRNA transcript, which we're going to represent as a red ribbon, threads through the ribosome. Okay? And there are three basic locations within the ribosome where tRNAs can bind. This first location, which I guess we'll do in blue, is called the A site. And A stands for amino acyl tRNA. So guess what goes there? The amino acyl tRNA. And how does the correct amino acyl tRNA fit there? Well, it's got to have the complementary codon, right? The anticodon. So, for example, if this is a phenylalanine tRNA, and let's say the sequence of the mRNA sequence is read by the ribosome from 5' to 3'. So a phenylalanine codon, as we said, would be UUC. Oh man, errors persist. UUC. So at this point, the mRNA transcript from, 5' to 3' would read I did it again, C for phenylalanine. And then the anticodon segment on the tRNA would read AAG from the 5' to 3' in that direction. Okay? So the right codon fits in the amino acyl site. And I think this is mostly just random. I don't think there's anybody whose job is to take the right tRNA over. I think tRNAs just go in and you sort through the 20 possible tRNAs until you find one that sticks. It's incredible that that can happen that fast, because protein synthesis doesn't take a very long time. And we could learn about how long it would take to make a protein from an mRNA, but your body's doing this constantly. So this is a very fast process. Okay, second site is called the P site. And P could stand for previous, but it actually stands for peptidal tRNA. Okay, so this is kind of cool. I should have, if I were smart, we should have colored the tRNA that holds phenylalanine. Maybe we should have colored it blue. That would have helped, maybe. Okay, I don't know. There's an A in there that's bothering me. All right, so in the P site is the tRNA for the previous amino acid, the one that was put on before. So in our sequence, let's suppose the previous amino acid was, I'm going to need a genetic code so I can get this right. Let's suppose that the previous amino acid was isoleucine. The three-letter code for isoleucine is AUU. That would mean the tRNA has from 5' to 3' the sequence AU, whatever. So this one's going to have attached to its 3' hydroxyl not only the previous isoleucine, but also the rest of the protein. In other words, supposing that all the way down to the original methionine, Suppose from here to here is 15 amino acids. Then this tRNA at the P site contains the elongating peptide chain. Does that make sense? Then how do you form a bond between the A site tRNA amino acid and the P site tRNA amino acid? That's going to be pretty simple. So let's draw how that happens. I'm going to use the letter P to abbreviate the P site tRNA. And here is the 3' hydroxyl on that P site tRNA. Here's the isoleucine side chain. And then here is the rest of the peptide that's being synthesized. The P site tRNA is going to be the electrophile. The A site tRNA amino acid is going to be the nucleophile. So this is the one with the phenylalanine side chain, right? Okay. A base from the ribosome, we'll just call it the enzyme, is going to remove a proton from the ammonium group. And as it gets removed, it's going to attack the carbonyl carbon and break the pi bond of the carbonyl carbon at the P site tRNA, thereby leading to a tetrahedral intermediate that's going to look like this. So here's the isoleucine with the rest of the peptide. Here is the new bond. I'm going to make it a little bit longer so that we don't run out of space. This is the new bond with the amino group on the A site RNA. And then this is the A site RNA. Does that make sense? Amino group attacks carbonyl carbon, get a tetrahedral intermediate. At that stage, a base will remove a proton from the positively charged amino group so that it doesn't leave again. So deprotonate. And all right, that will get us to this intermediate. The carbonyl carbon, it's a tetrahedral intermediate at the carbonyl carbon of the P site tRNA. I'm just going to use R for the rest of the peptide. The amino group is now not a good leaving group. We'll make this the A site tRNA. Now electrons on the negatively charged carbon are going to kick down and they're going to kick off the P site tRNA as a leaving group. It will pick up a proton on its way out from probably the conjugate acid of the base that we used. And what we're left with is now the P site tRNA no longer has the peptide. Its 3' hydroxyl group is free. That was the leaving group. And now the A site 3' hydroxyl group has now phenylalanine but that phenylalanine is now bound to the isoleucine, which is now bound to the rest of the peptide. This is the N-terminal side of the peptide. This is the C-terminal side of the peptide. Ribosomal protein synthesis inserts an amino acid at the C-terminus. Okay? So if you're following along after all of this, after this step, we should have the P site tRNA no longer bearing the peptide. And then you should have the A site tRNA now bearing all of this, which I'm going to sort of copy and paste. Sorry. Close enough. All right. Now, in order for it to add a next amino acid, the ribosome needs to translocate down the chain. It needs to move the P site RNA to this position, which is called E, and E is for exit site. So what ends up happening is the P site tRNA gets, this is going to be dumb, it's not really good animation, gets translocated here to the exit site. And then the A site tRNA with all of its, this is going to be terrible. The A site tRNA with all of its baggage gets translocated there. And I didn't mean to move everything. And now the A site is free and ready for another amino acyl tRNA to come in. Okay, so it's just amide bond formation. This is the amide bond we just barely made by the ribosome. Notice that we started with, if you come back all the way here, we started with an ester functional group. That's level 2. We replaced that ester with a more stable amide functional group. That's level 3. So this process is downhill. Okay? I'm not so concerned that you memorize. I really have almost no interest or time in asking you questions about A site versus P site versus E site in the ribosome. But I do want you to know the mechanism of amide bond formation because that's not really new. You've seen it before. The players are just different. I also wanted to show you, and I know we need a break, but this is too cool to wait. I wanted to show you a movie from a GIF on Wikipedia about translation to give you an image of what's going on. Oh, it must have been up at the top. I hope. There it is. All right. So what you're seeing in this GIF, the dark blue things are tRNAs. The light blue things are what are called elongation factors that actually help bring tRNAs to the ribosome and then push them through. That translocation that I told you about where a tRNA in the A site needs to move to the P site, there's literally something that comes along and pokes it down the line. As this happens, the peptide chain, and here you're seeing this is even better, the assembly of the ribosome. As A sites go to P sites, you're seeing, and I need a laser pointer, this is the elongating peptide chain. A site now to P, and the chain just gets transferred from one tRNA to the other as this elongation factor sticks in and pushes it down the line. The peptide emerges through a tunnel in the ribosome out here, and you can sort of count, there's probably 10 to 15 amino acids that are embedded in the ribosome before things continue onward. If what you're seeing here is if the peptide has a specific amino acid sequence, there's actually a factor that delivers the ribosome to the endoplasmic reticulum and inserts the peptide through a portal into the ER for further processing. So nature's like crazy. But the important thing that we're talking about here is tRNAs deliver amino acids and then the peptide gets synthesized. Kind of wild. Okay, anything that you want to know? And don't be worried at this point. I think I told you what I want you to know for the test. So if you're curious, don't be worried that you're going to ask something and then I'm going to be like, ooh, that's a good test question. You don't believe me though. Go ahead, what do you think? Questions about this? Okay, well, you'll learn more about that if you want to. Let's take a break, and then when we come back, we're going to start in with beta-lactam antibiotics and NSAIDs.